What does a biostatistician see when he looks at cancer vaccines? Let's find out from someone right at the center of it all.
Dr. Gönen has spent over 25 years at Memorial Sloan Kettering, designing and analyzing clinical trials across chemotherapy, targeted therapies, and now immunology. His entry into cancer vaccines came through a collaboration with Dr. Vinod Balachandran, studying neoantigens from tumor DNA and building a phase 1 trial on BioNTech's platform that was safe enough and promising enough to move to phase 2.
Here's a distinction worth holding on to. Traditional vaccines prevent disease before it starts, but cancer vaccines are treatments given after diagnosis, designed to help the immune system recognize and attack cancer cells.
In theory, a cancer vaccine works along 3 lines: shrinking existing tumors, killing cancer cells, and stopping them from coming back, making it firmly a treatment, not a prevention.
To understand where cancer vaccines are today, it helps to look at why their development has been such a stop-and-start journey over the past 4 decades.
Early cancer vaccines borrowed methods from infectious disease research using peptides, and they failed. Badly enough that many researchers wrote off the entire field until advances in molecular biology showed a completely different path forward.
That loss of faith now looks premature because researchers believe they finally have the right technology to make real progress, putting the field at what feels like a genuine turning point.
2 scientific breakthroughs in particular have driven the recent momentum in cancer vaccine development, and they work hand in hand.
The first is the ability to study tumor cells with far greater precision, understanding how they differ from healthy cells and how they hide from the immune system. The second is the ability to actually make sense of the millions of data points that precision generates.
Molecular biology, computing power, and statistics have all advanced together, and their convergence is what's giving researchers a real shot at waking up the immune system to fight cancer.
Cancer vaccine trials have faced real hurdles, and it's worth separating what we know from what remains genuinely uncertain.
Phase 1 trials have consistently shown cancer vaccines to be safe in the short term, but long-term effects are still unknown and will only become clear as trials mature over years of follow-up.
One of the deepest open questions is whether a single vaccine approach can work across different tumor types with minor tweaks, or whether every patient truly needs a vaccine built from scratch. And right now, neither answer has been proven.
So what exactly is a personalized neoantigen vaccine? And why does the science point toward individualization in the first place?
Neoantigens are proteins that appear uniquely in cancer cells, produced by tumor mutations, and they act like flags that the immune system can potentially learn to recognize as a sign that a cell is cancerous.
Because every tumor has its own neoantigen signature, a sample is taken from the patient's tumor, sequenced to map those specific proteins, and used to build a synthetic mRNA vaccine tailored entirely to that individual.
The catch is that tumors express thousands of neoantigens, but a vaccine can only target a small subset, so identifying the right ones, the ones that will actually trigger a strong immune response, is one of the hardest problems in the field.
Once that personalized mRNA vaccine enters the body, it signals the immune system to recognize cancer cells as threats, activating T cells and natural killer cells to mount a coordinated attack.
This approach has worked best in patients who had surgery, because a surgical specimen provides far more tumor material to analyze, directly linking the quality of the tissue sample to the quality of the vaccine.
With thousands of neoantigens to choose from, machine learning algorithms have become essential for narrowing the field down to the ones most likely to matter for each patient. These predictive algorithms look for neoantigens likely to trigger tumor-infiltrating lymphocyte responses, a signal first spotted in melanoma that correlates with tumor shrinkage, though false positives have been common, with many candidates that look promising in small samples failing to deliver in the body.
The algorithm isn't just a behind-the-scenes tool. When a personalized cancer vaccine goes to the FDA, the neoantigen selection algorithm is expected to be part of the submission, reviewed much like a software device.
One of the most revealing early tests of this approach came from a phase 1 trial in one of the hardest cancers to treat, pancreatic cancer, run at Memorial Sloan Kettering with BioNTech and Genentech.
Pancreatic cancer was chosen for its poor prognosis and surgical accessibility. 18 patients enrolled, and personalized vaccines were successfully manufactured for 16 of them using BioNTech's platform.
Of those 16 patients, 8 mounted a strong T-cell response to the vaccine's neoantigens and were called responders, while the other 8 showed no such immune reaction. A clear biological split with striking clinical consequences.
Over roughly 2 years of follow-up, none of the responders experienced tumor recurrence, while all but one of the non-responders did. A small but compelling proof of principle that the right neoantigen targets can genuinely hold cancer at bay.
That surge in neoantigen-specific T cells after vaccination looks like a promising biomarker, similar to neutralizing antibodies in infectious disease vaccines, potentially allowing researchers to predict long-term outcomes without waiting years for survival data.
Delaying recurrence isn't just a number on a chart. In pancreatic cancer, where recurrence brings severe nutritional problems and crushing fatigue, buying more time before relapse is a direct and meaningful improvement in a patient's daily life.
Moderna and BioNTech are the two frontrunners in personalized mRNA cancer vaccines. And while they share a lot of common ground, their strategies diverge in one important way.
Both use mRNA and pursue individualized neoantigen vaccines. But Moderna pairs its vaccine with checkpoint inhibitors like Merck's pembrolizumab, while BioNTech leans toward combinations with chemotherapy or targeted agents. A difference that reflects each company's partnership structure as much as its science.
Step back and look at the full landscape, though, and these two companies land very close together. More alike than different, and clearly ahead of everyone else in the field.
Looking ahead, the question isn't just whether cancer vaccines will be approved, but what happens to the entire field once the first ones are. Dr. Gönen draws a direct parallel to checkpoint inhibitors a decade ago. One approval led to a second, then suddenly thousands of trials opened up, and he expects cancer vaccines to follow the same explosive trajectory once the first 1 or 2 cross the finish line.
Funding is already building from every direction. Pharmaceutical companies, individual donors who have been personally touched by cancer, and government agencies like NIH and NCI are all pouring resources into the field at once.
Payers and reimbursement authorities are also likely to warm to personalized vaccines because a therapy matched to each patient's tumor carries a stronger case for effectiveness. And that makes the economics easier to justify too.
That said, a note of caution. Personalized medicine was also the big promise 15 to 20 years ago with targeted drugs. And while those drugs helped, they were never truly individualized. Neoantigen vaccines have more potential, but history suggests keeping expectations grounded.
Among smaller players, one company stands out, Aston Sai, a Korean biotech that has already received FDA IND clearance for a vaccine candidate in breast cancer, separating itself from the broader field of early-stage firms.
As Dr. Gönen puts it, once we have 1 or 2 approvals, the floodgates will open, just as they did with checkpoint inhibitors a decade ago.